they were
Yes,
the
hello.
most educated bunch.
Good morning. My name is Fabrice Delay. It's a pleasure to start this press conference with this very prestigious panel of scientists. So you may hear I have an accent. I'm from France, and the reason I'm here is because about a year and a half ago, when the vaccine were authorized, the mRNA vaccine were first authorized to cure COVID, I asked myself a question, which, I mean, everybody was asking that question. I was so fast, and I tried to find the answer, and I enter in a work of reconstituting the 30-plus years of history that made possible this incredible technology, because it's an incredible technology. And I interviewed most of these gentlemen to write what was to be an article that finished as a book. And this book was noticed by people here, and including Douglas Freeman, which I'm going to invite to join me. So he invited me to try to make this story accessible for an even larger audience, that is you today, and that's the reason I'm here. So maybe Douglas, can you explain why you invite me, why you invite these gentlemen, and what the celebration of you?
Thank you, Fabrice. Ladies and gentlemen, members of the press, guests, this is a important day for us here in Orange County and in Southern California. It was an idea that occurred to myself and my colleague Charlie Zhang, and those of you who know Charlie and know his background, but Charlie was the Chinese immigrant who created the restaurant Pickup Sticks, and those of us who live in Southern California all made him rich. He's an amazing American philanthropist, and he and I decided that we needed to have a day of celebration, but it wasn't a day to have a party. We were in the middle of a pandemic. This is March of 2021. We decided if we're going to celebrate, we're not going to celebrate because we killed the virus. No, that's not the issue. We're going to celebrate because science and the scientists have done an amazing miracle. They created something over 3 decades that has saved a lot of lives. And we felt blessed. And the first thing that we wanted to do was to have this county recognize the work that the scientists and their colleagues have accomplished. So in addition to wanting to bring the scientists from around the world here, and there will be another group of scientists that we're honoring in addition to these gentlemen that are still in their labs, and we can't extricate them to come here, but they are in your book, and in your gala book, you'll read about them. But we decided that beyond the scientists, we needed to also recognize the men and women in healthcare, the men and women in the senior centers, our teachers, our grocery clerks, our ambulance drivers, God knows who. We needed to say thank you because they were making our life possible. They were saving our lives, they were nurturing us, they were giving us hope, and they were giving us friendship at a time when we were all huddled in our bunkers. And so we have spent the last year looking for these heroes. You're going to meet a few of them. One is here this morning, that young lady, Patricia Gallagher, who we brought here from Philadelphia because of the work that she has done. Then you will meet others here today, both in this conference as well as in the panel discussion. Those men and women were essential to our well-being throughout Southern California and throughout the country. They represent a much larger group of men and women and young adults who made life possible for us again. So it is a day of celebration, but it is a day to celebrate our gratitude to these men, their colleagues, their lab assistants, and all those upon whose shoulders they stand, together with all of our local heroes, heroes we call the heroes of the pandemic. So while you're here today, and you're going to get a good insight into the science and where the science is and where it may be going, remember those heroes that also took care of us when we got sick and kept us out of the hospital and brought us back when we thought we were going to die. Those people need to be thanked as well. So thank you all for coming. We hope this is a very meaningful and insightful day for you. And I am very pleased that you're here to meet these extraordinary men. Thank you so much. Thank you, Fabrice.
Thank you, Douglas. So, yeah, maybe let me do a very little quick introduction about what messenger RNA is and why this technology matter at that moment. And then I propose that you will ask your question about maybe the past of the technology, because all these people here have been pioneer. I'm going to introduce them to you in a minute. And maybe about the potential of this technology now, be it for the pandemic, pan-coronavirus vaccine, for example, other technologies that are possible for other infectious disease, for cancer, for genetic disorder. I mean, the potential of this technology is quite unbelievable. And the reason why, it's because you have to realize that it all started in the 50s. In the 50s, there was two major discoveries in biology that were made, one by a gentleman named Fred Sanger in the UK, who basically understood that proteins, which is the building blocks of everything that is life, everything that is acting in our body, it's proteins. He understands that there's a certain sequence of organization in the biochemistry of that. And 3 years later, in 1953, Watson and Crick discovered the DNA, which is the memory. And the question was, how do we go from this information, I would almost call it a cloud today, of information that is in the DNA into something practical, something acting, that is the protein. And in 1961, a group of French and American researchers, both at Institut Pasteur and here in California, says it's the messenger RNA. That's that thing that is transforming this information, this universal information, into something acting in the body. And it could be anything, insulin, growth hormone. I mean, the number of protein is endless. And so after the science developed, and at a certain point, and that's where we are going to start, this science became applicable. It became the basis of medical application of technology. At the end of the 90s, that's when pharma company and biotech companies start to show their interest in the beginning. So 30 years ago, and but it was not finished. There was the idea, but the process to develop that, these gentlemen are going to explain you what they've done was very long. So if I'm trying to put a little bit of order in all that, I would say that, where is Walter Shaw? Yeah, Walter is here. Walter, Phil, maybe you can raise your hand. So Walter Shaw, Phil Feligner, and Peter Kulis, they have developed, it's two technology you have to understand. It's mRNA and it's what we call now lipid nanoparticle. At the time it was called liposomes, which is the delivery mechanism. So mRNA is like an information technology. It's the code. But you need to bring that code into the cell, and it's not trivial. These three gentlemen developed this technology, and I would say Walter Shaw was a pioneer of liposomes. Phil Feligner here he invented what they call the cationic lipid. So he's using electrical property to help the delivery. Peter Kulis even developed more on that because there was toxicity problem, and this is the basis of what is today the one big block of the technology, the lipid nanoparticle. Then you can ask also your question about more on the immunology, the vaccine discovery, and then I would say that Drew Weissman, maybe you can raise your hand, Drew, who is a professor at University of Pennsylvania. Jeffrey Helmer, also an immunologist. He has been a very key moment in the different time of the history of the mRNA technology. I would say, Barney Graham and Jason McClellan. They have been also working more on the, if you want, the molecular biology side, or more on the mRNA side, and even more of the protein side. Which protein do we want to design that will be efficient for a vaccine? So that said, I think I have not forgotten to present anyone. Henry he is the president of the American Society of Clinical Pathology, right? And he has been also working on the side of, on a very important side, which is testing, which is identifying that damn coronavirus. So you can also answer to your question about this important field. That say, I think I leave it for you for your question. You can tell me whatever question you have, and I will try to help you to identify who is the best person to answer them, if it's okay with you. So, any question? American Journal is not asking question.
So my question is, work in a silo in the fields that you're in. You can't work in a silo in science. So I'm interested, from your experience, how did you interact with each other? Because science is not like a business, a traditional business where they protect every ounce of proprietary information. Tell us a little bit about this process that you went through in those early days and maybe in the different stages to begin to interact with each other, because you all did. And I'm interested in how was that done, and what was that experience like?
Maybe we can start this one with Jeffrey and Philip, because they have been collaborating in the early 90s on the development of a first vaccine candidate.
Actually, I think that we got involved even before we got involved in an agreement with Merck, Sharp and Dome. But prior to that, the liposome field was very collegial, and we had very active meetings, and everybody was sharing everything they knew. And it wasn't a company consideration of patentable technology that we had to preserve, keeping things confidential. It was more that we had to figure out what we needed to do to get something done. And remember, the liposome company had these amazing meetings that they put together every year with lots of scientists from all different fields, and so we were learning so much. It wasn't until later, when you start actually being involved with a company and there's intellectual property that has to be protected, then that's a different ballgame. And I think that we learn about that as scientists just from experience. You know, at first it's real science and it's collaborative, but then when there's some intellectual property of value, then there's a kind of a shroud that enters in. So it wasn't really a problem at the beginning.
Very few successes that I think happened truly in isolation. In the case of the collaboration, I guess, started about 19. Merck, when I was at Merck, Bill, when he was at a company called Vical in San Diego. What we at Merck were attempting to do was to make a vaccine that could do some response that isn't typically by the vaccines that are out there. They're called cytotoxic T lymphocytes, and there are cells that survey and look for virus infections and then kill those cells, and that they facilitate the clearance of the infection and hence. But it wasn't easy to envision that with a vaccine. We heard a rumor, and I'm not even sure how we first found out, that you could simply take genetic material, be it RNA or DNA, encoding the antigen you want to target the immune response against. Inject it, it gets into a cell, it expresses protein, and kind of mimics a virus infection. And in doing so, then it can, in a very facile way, generate this kind of immunity. Contacted them, and it started a very fruitful collaboration. We tested probably infectious disease targets with DNA and RNA. And in fact, one of the very first experiments we did was to compare head-to-head DNA versus RNA. And showed that both of them worked very well. The focus initially in that time ended up to be on DNA because at the time we didn't see a clear path forward to industrialize an RNA-based product. But fast forward to today, and those issues have been solved. Productive collaboration where both parties brought something to the table.
And I would say, maybe I would ask the same question about the importance of interaction about two persons that have been key into scientists, but key into the industrialization. That is the interaction between Drew Weissman and Peter Kulis. That maybe you can explain why this collaboration started.
So Katie Carrico and I have been and continue to work on RNA for almost 25 years. And then in 2005, we discovered nucleoside-modified RNA, which is the RNA that's in both the Pfizer and Moderna vaccines. But soon after that, and even before, the issue came, how do you deliver it? Because RNA is degraded the minute you put it into a body. So we were looking at ways to deliver the mRNA as vaccines and as therapeutics. And Peter and his collaborators had developed cationic lipids with specialized function. So I spent a few years trying to get that from them. And sorry, that always but
wasn't my fault.
But Peter and I connected, and I connected with Acuitas, which is a company he started, and we started working, and the results were fantastic, and we just kept going from there.
Yeah, just to emphasize the collaborative nature of things, but also the history here, which Phil was also referring to. We all started, I mean, you're looking at a bunch of old white guys here, right? But we all started long ago in a field that wasn't exactly, center stage, let's put it that way. And so the part of that was we sustained ourselves, maybe it was 100 people or something in that range. With these meetings where we met. This goes on in science all the time. There's specialized areas where people with particular interests will congregate, have a convention or whatever, symposia, and it just slowly grew. And so of course, then when you want to get something into the clinic, you want to actually make a drug out of something. That's when we got drawn into the company side, because there's no way you can do that in an academic setting. You just need too much money to do the clinical trials and all of that. So I got involved with starting a company, and then this was to deliver cancer drugs more specifically to tumors. And then one day in the middle 90s, the CEO came to me and said, I can't raise money putting these old drugs into liposomes. We got to be doing gene therapy, or we got to be doing something with DNA and RNA. And so this was a huge challenge, but it ended up with then we switched into that field, and it ended up with some of the systems that are now, they were the precursors to the systems that are used vaccines. So this is the way science goes. You have the very basic stuff being done, and then there's translation, and that requires a different mindset. And then you get these happenstances where we were going in a direction that had really nothing to do with vaccines, but Drew recognized it could be useful in a vaccine application. Calls us up and says, "Let's have a go at this." But you couldn't prophesy these things. Science is a very nonlinear process.
And maybe a last example of interaction that has proved key is also the one between Barney Graham and Jason McClellan, their collaboration. I mean, there would be no vaccine if they have not been collaborating on a very, very clever idea, as they are going to explain to you. Maybe you can explain what was the work and how you collaborate and your interaction.
Yeah, first of all, I want to just say that who you see up here is a very small part of the teams that have. Even among the people being honored, you're only seeing part of them. So, for instance, Dr. Kizmikia Corbett, who was a fellow in my laboratory, is now starting her own lab at Harvard. She's a new assistant professor at Harvard and just at the verge of getting her lab opened, hiring people, and so she wasn't able to make the trip, but I wish she had been able to be here. And I know Caitlin Perico wasn't able to make it. So first of all, it's just there's a lot of people involved, and we're representing them. I come from the Vaccine Research Center. It's a program that was started on the intramural campus at NIH back in the year 2000. So it's been there about 21 years. It was conceived around 1996, 97, and it brought multidisciplinary scientists together, going all the way from very basic structural biology through clinical trials, and housed us all in the same place with the mission of making an HIV vaccine. This process of bringing different fields together helped us exploit technologies from different fields, from different perspectives, to put together not a successful HIV vaccine, which we still don't have, but the technologies derived from that have been exploited now to make other vaccines, including contributions to this coronavirus vaccine. For Jason and I, in particular, Jason was there as a young postdoc to study HIV structure, but the lab on the fourth floor was so successful, he had to find room on the second floor where I had a lab that was studying a different virus, respiratory syncytial virus. And so I attribute most of my success to the day that Jason came down to the second floor and to help me with a different virus. And from that, we learned about how capturing the right structure of the surface protein of the RSV, an envelope virus similar to coronavirus, all of a sudden changes that protein into a very good vaccine antigen. And so extending that observation to coronaviruses is how Jason started his faculty position at Dartmouth back in 2013. And working together with him, some of the information we needed for coronaviruses had been developed over these last 8 years, just in time for this pandemic to occur. So there's been several threads of other viruses, other technologies, mRNA, lipids. There's a lot of threads that have all came together just at the right time for this.
I think Barney really nailed it. What we focus on is trying to choose and optimize the best antigen, the best part of the virus to use as a vaccine. So it's somewhat agnostic to the delivery platforms. We came up with a method of making changes to the coronavirus spike protein, also the RSVF protein, that make it a better vaccine antigen. But that can be delivered by mRNA, also by adenovirus, DNA, or just this purified protein. So the stabilized form of the coronavirus spike protein we've developed is used in both of the mRNA vaccines, but also in the Johnson & Johnson adenovirus-based vaccine, the Novavax protein-based vaccine, Medigen's vaccine in Taiwan, and several second-generation ones that are coming online now. So it's one component. When you make a vaccine, you have to choose what part of the virus or pathogen do you want to present to the immune system and how best to present it, and that's what we do.
Yes, Miles?
So a question for you about the extraordinary timing and synchronicity of all of this. You guys are overnight successes after 30 years of work, right? So 30 years of toil and all of a sudden this extraordinary 10-month sprint with an existential crisis. I'm curious how your work changed in that 10-month period, aside from the simple answer that you went faster. And have you ever thought about the hypothetical of if you hadn't had this pressing deadline facing you, where would your work be today? Would you still be waiting for approvals, or would you be further along?
Maybe Barney can start with that because it starts with the sequence that came from China.
Right. The history of vaccines is really all about taking opportunistic use of new technologies to advance a new vaccine. That goes all the way back to when Goodpasture learned how to grow viruses in eggs. That made the yellow fever and first flu vaccine possible back in the 1930s. So what's happened over these last several years is that as we got structural information for the coronavirus, learned how to stabilize the coronavirus spike as an antigen, and as we failed to respond fast enough for the West African Ebola or the Zika in 2016, and develop better ideas of how to be better prepared for pandemics, and as the mRNA technology came on board, we had started a collaboration with Moderna in 2017 for something called the prototype pathogen approach for preparedness, which means you select a virus representing a group or family of viruses and try to figure out interventions for that particular virus, hoping you can generalize it and apply it to the other viruses in that family of the 26 virus families that are known to infect humans. And use that as the paradigm for being prepared. So we were working on paramyxoviruses and coronaviruses with Moderna since 2017 with the plan that we would design antigens, they would deliver it with mRNA, and we'd combine precision with speed, and that would be our pandemic preparedness plan. And working on Nipah virus and MERS coronavirus is the prototype. Had a plan in the first quarter of 2020 to take our Nipah product into the clinical trials, because we'd shown both spike for coronavirus and the FG chimera for paramyxoviruses could work by mRNA in animal models. And when those sequences came out on the 10th of January, learned it was beta coronavirus around the 6th of January of 2020. I talked with Stefan Vancel at Moderna, and we said, well, instead of paramyxoviruses, I think coronaviruses should be our demonstration project. So this was really started as a demonstration project, not a response to a pandemic. Didn't have a case of infection in the United States until after we started. And the pandemic wasn't declared until really right at the time we were starting the first phase 1 clinical trial. So this was a confluence of events that you couldn't have imagined, as Peter was saying, that it all came together with all the right information and all the right tools from a number of different threads, and was being executed as a demonstration project, not knowing that it would necessarily turn into a pandemic. The point was to go as fast as you could, because that was the demonstration for pandemic preparedness. So there's a lot of very fortunate things that were lined up right at the right time with the right knowledge in place to make this happen.
One of the key factors in speed, fundamental difference with the mRNA technologies compared to traditional vaccines, and that is all the vaccines that we used leading up to coronavirus are made in biological systems like cell cultures, big vats of liquid where you're growing cells. Make the mRNA and lipid nanoparticles completely synthetic, complication, but more importantly, it creates the potential for a true platform technology, whereas once you've identified the methods and means to characterize them, you can copy and paste them from one product to the next product. And so I think that is one of the key enabling technology features of this that has made it possible to go faster.
Well, Avani, my company, is a lipid producing company, and we had the capability of making these compounds under the guidelines of the good manufacturing practice, which made them available for clinical use. And so we had all the components for other projects that the lipids were available. But we were a small company in Alabama, and to supply the world with the lipids, we needed some large company. And we went to Croda, which had capability of laboratories all over the world. And we were able to transfer our technology to Croda to make large quantities of these lipids for the nanoparticle and do it under CGMP. That was kind of a miracle.
Henry, in a different field, which is testing, but you also have an experiment of that speed and this feeling of urgency, and maybe you can tell us about a little bit about your experience.
Sure, thank you. We'll have to sort of take a little bit of a switch away from vaccines and talk more about testing for the RNA of the virus. But a lot of the situation that you described is parallel. We didn't know we were going to have coronavirus in January 1st of 2020 that we had to test for. But for many, many years, laboratories where medical technologists and pathologists work, they have to build tests from scratch all the time. Tests may not be available from commercial sources. They may be too expensive. They may not be as good. And so laboratories are actually used to developing their own tests, not with big R&D teams, but with the technologists that they actually have on hand who are doing their work on a day-to-day basis. They build, design, validate, and then implement the tests under the inspection of FDA or accrediting bodies. And so you can imagine here we are in early 2020 where our lab and many others, and as Barney says, I'm just here to represent the lab techs and the pathologists and the laboratorians that did this. No one has a sample of virus. No one has a patient who's been infected. We don't have any live samples from the nasal pharyngeal area or the oral or the lung. We have nothing. But because of all the collaboration of years and years from investigators, we have the sequences. They're available on WHO. They're on GenBank. And of course, you have all of the virology laboratories talking with each other. In my institution, Marie Landry was a highly accomplished international virologist who had connections all over the world and in FDA. And so they're all talking to each other about what are the best, not targets for vaccine, but targets for detection. And so the different proteins, envelope, spike, et cetera, are all actual targets for us to be able to detect. The FDA, right at the end of February, the first positive cases were in California and Washington, opened the door to the laboratories and said, you can go ahead and develop your own tests. And they gave us a very large template that we had to follow and be able to document, that they would then give us approval, an EUA, if we were able to follow all of that and get good testing done. So starting on March 1st, an all-out effort, 24/7, every single day, nights and weekends, lab techs who are working their regular job, do double and triple shifts to try and figure out the best way to test for the virus. And the first week of March in Connecticut at my lab, we actually got our first cases. So now we had actual clinical samples. And the same thing's going on all over the country. In California, in Washington, where Jeff Baird was, the Cleveland Clinic, where Gary Prokop is working. We're all doing this. We can't rely on one lab because if they don't make it, then we've put all our eggs in one basket. So we're all working together, but all talking about this to try and figure out the best things. And every component of it, the swab, the media that it's got to go in, the primers. We wish we had a test where we could take the swab and stick it in a black box and the light would go on red or green. Don't have that. Every single aspect, getting the RNA out, amplifying it, finding the right level to be able to say it's truly positive and is not cross-reacting with a different coronavirus like MERS, as you were discussing. So in 2 weeks, our laboratory went live with testing and kept on going with the data such that by the end of March, we submitted and got EUA approval. We were the first laboratory in the country to get EUA approval for a high complexity SARS-CoV-2 mRNA test. And this was being replicated all around. And you might think, okay, well, great, you've got this going, and now you're done, right? You've got a test. Now the companies will come pile in and we'll all be working together. But now the supply chain problems occur. So you got the test going, but all of a sudden your viral transport media goes away. Well, you can get substitute viral transport media, but now you must revalidate the entire test with a new component. The commercial companies are coming along, and they were very close behind with us to help with that. But by the end of the time period, acutely, we had validated 16 SARS-CoV tests in our laboratory, many of which were operating simultaneously, depending on their turnaround time, to be able to make sure that patients who were coming in who were acutely ill or patients that were coming in for elective surgery could be screened to protect our healthcare workers so that they were known to be negative before they went in for surgery. So it echoes a lot of the principles and the collaborations that these distinguished scientists have talked about. And this was all over the country, in every academic and other laboratory, and in the industry that was developing these tests as well.
Thank you. Is there any question? Yes, please.
Thank you so much. I'm so impressed with everything. Was there ever a time that you felt that your lives were in danger working with the viruses as you were developing the vaccines?
Who wants to take this one? Barney, you want to like?
I'll start and then Jason can finish. But
Okay.
Interestingly, at our lab at the VRC on NIH campus, we never had the virus. All we needed was the sequence to do what we needed to do. And we could build all the reagents and everything we needed without the virus present. The part that was hard was that once we got to March, we were under restrictions for workforce restrictions. We had to keep our students and people in the lab separated. We could only have about 25% or less of the people we're used to having in the lab in the lab. In fact, at the VRC, it was the only building on NIH campus that was actually open for research. Everything else, we were at about 20 to 25% capacity trying to get the work done to keep things moving. That was really the biggest problem. And so I never felt in danger, but I worried about the students and the young people in the lab and the technical people and the other scientists that were helping to get the actual hands-on work done because they had to travel. Some of them were on public transportation. There was a few infections early on that shut the lab down for a couple of weeks. And so that was the hard part, was that early phase where we couldn't really, in a certain way, keep the work going. And so a lot of new strategies had to be developed with multiple work sites and different shifts and things like that. We never had the virus in the lab.
And we can just go from digital sequence posted online to making DNA in the lab, put it into mammalian cells, have it create viral protein, but that's not infectious, it's just one protein of many. Then we can structurally characterize, modify, tweak it, but we never need the virus. We collaborate with people who do, Ralph Barrick at UNC and others, Mark Dennison, in order to test these with authentic virus. But really early on, throughout the last 2 years, I think the healthcare workers have just been incredible heroes. They've really put their lives on the line. They've been working with people, patients who are infected, being exposed to large doses of the coronavirus. Many of them have lost their lives. And so we were relatively safe in our labs, but the healthcare workers were really on the front line.
I would echo that. And we were the healthcare workers in the laboratories. So we were actually multiple weeks into testing with live virus. We're at a meeting and we were having discussions about this, and one of our head people in the hospital said, and you guys are all protected, right? You have personal protective equipment. And we said, no, the laboratories don't have personal protective equipment. So right then we got, in about 10 minutes, we got 100 boxes of PPE. We, thank God, we were lucky. We did not have anyone in the laboratory die, but we did have many people get sick. And in fact, this was actually a barrier to doing autopsies. 150,000 people worldwide died, and at that time there were only 15 reported autopsies. That's 1 in 10,000. So there was not enough knowledge out there about the clinical illness and the pathophysiology of the disease as well. Luckily, over time, people realized they could protect against transmission. The virus is alive for at least 6 or 7 days post-mortem, and so there were precautions that were taken for that.
Good morning. My name's Craig. I'm a reporter with KNX. And a couple of things for my coverage, and also whoever wants to answer this, when you do, if you could say your name first so I know who it is. I've looked at your pictures, but some of you look different. Not in a bad way, not in a bad way. I wanted to know, early on in January, I talked with Dr. Nancy Messonnier, who was with the CDC then several times, and we did stories on, and I know one of you said that it wasn't declared a pandemic, but in January it certainly seemed we were on that path. So I talked with her about China. Is China giving you good information? As I think the term I use, are they giving you the straight dope on what's happening on the ground there? And she's kind of hedged and gave me an answer, non-answer. I want to know if what was happening in China, what was coming out of and not coming out of, helped or hindered your research for mRNA. And then my other question is, with mRNA now having been rushed or forced to the finish line in a good way, in a real world, event, what is the future of mRNA for other ailments?
I think the first question is Barney to answer, because you've been, you have been pressuring your colleagues at the time, if I remember correctly, to get this sequence. It was not that easy to get it at the first. I mean, apparently the story is a little bit twisted.
No, in those early days, it's pretty chaotic, and you don't really know, how it's all going to play out. But I really think people were doing the best they could with, in the settings they were in. And so, what we needed was the sequence to get the program launched. That's really all we needed. And so, I did have some communications with Dr. George Gao, who's the head of the China. Back and forth a few times. Dr. Fauci was involved in some of those communications, and that morning of January 10th, I did send him an email and said, when are we going to have these sequences? Because it's not just your problem. It's going to be everyone's problem. And said he's working as hard as he can to get them out. And
Didn't really answer you, as I recall. He kind of dropped off the radar at some point.
I know that he was trying to get them out, and later that night they came out and were posted. Now, I don't know that I had anything to do with it, but I know that they were working through the bureaucracy and all the political systems that they have to deal with to get those sequences out. I trust that they were doing the best they could.
And my second question, again, for whoever, what is the future of mRNA success in curing or dealing with ailments in humans, given this success with COVID-19, SARS-CoV-2?
Yes, well,
Yeah,
all of them
I think-
can take it, but maybe we can start by COVID to start with, variants and pan-coronavirus vaccine, and then move on to the other infectious disease, and then cancer and other, because the technology is pretty wide. So who wants to start with pan-coronavirus?
So
Drew?
yeah, the future of, I'm Drew Weissman. The future of RNA therapeutics is enormous, and this is what got Katie and I started in 1988, that we saw enormous potential for this kind of therapeutic. Where we are now, we and others have made pan-coronavirus vaccines. We have universal influenza vaccines in clinical trials. We have HIV vaccines in clinical trials. We have malaria vaccines headed that way. So there's enormous potential for vaccines. I think what's even more exciting are the other therapeutics. So we published a couple months ago that we could make CAR T cells in vivo. What that means is that CAR T cells are used to treat many different diseases, mainly liquid cancers right now, leukemias and lymphomas. It's a very complicated procedure. It's $500,000 per dose in cost because you have to do everything in a cell culture facility. We're able to make CAR Ts in the body. We inject RNA LNPs, and an hour later you have CAR Ts attacking whatever they're directed against. We have developments for gene therapies for sickle cell anemia, where we can inject RNA LNPs that go to the bone marrow stem cells that produce red blood cells and fix the beta hemoglobin gene to cure the disease. 200,000 people a year are born with sickle cell. So the only way to treat them is with a simple injection.
Maybe, yeah, Jeffrey?
Likewise, I'm very positive about the future of mRNA vaccines. I think it has the potential to address a variety of unmet medical needs that so far our existing vaccines technologies have not been successful. We demonstrated and validated the speed with which you can respond, and we've only learned from the COVID experience and will be better the next time around. There's also a fundamental attribute of the technology that I think will really have a big impact on vaccines research and development. I said, it's platformable, meaning you can copy and paste from one product to the other. That's not the case today. Every new vaccine product, even if it's based on the same technology, has to undergo all the same product-specific testing and characterization, which can take years and really adds to the lifetime of the R&D program. We can establish this as a true platform technology that is effective against other targets. It will have a significant effect on reducing vaccine R&D timelines and costs. And more importantly, it's going to get vaccines to people who need them.
Another aspect is in personalized medicine. Our usual track to developing a new medicine involves many years and many dollars to get to a drug that might be able to treat a particular disease. The advent of the mRNA approach allows you to develop a lot of these medicines in what you might call real time. So say an individual has cancer and there's a particular feature of that cancer that you want to target your medicine, you target an antibody to. The potential is there now to be able to detect what that epitope might be, that marker, and make an antibody against it, design an antibody against it, and then have your liver have a LNP mRNA system to have that protein made in situ in your liver. And so this highly personalized approach that can be done in a time on the order of maybe a few weeks. This is an incredible thing that these personalized therapies are going to really revolutionize medicine, in my view. We're all individuals, and as we get older, we get more and more individual. And
Yes.
So we need medicines and approaches that really cater to those differences.
Bonnie?
I just want to add one thing about mRNA, and there's been something that's confused a lot of people, I think, because it feels like such a mysterious new thing. But mRNA has properties that not only help make a more authentic immune response, but it is so simple. It is the simplest way of delivering a vaccine. mRNA and the lipid itself are gone within a few hours. The thing that's left is just the one protein you want to target, so you don't have to use the whole virus. It's eliciting all the right immune responses, and so if you are thinking about simple and elegant and probably as safe as anything could be, because the chemical synthesis allows you to control exactly what the content is. So you've got something that goes in, it only goes into the cytoplasm of the cell. It doesn't go to the nucleus where it can affect DNA that a lot of people have gotten confused about. And so it's simple, it's elegant, it's safe, and it's gone in just a few hours. And so I just hope people can start getting that message of how the mRNA actually works and how it would compare to other more complicated processes.
Any other question? Yes, please. Oh, sorry.
Hi, good afternoon, evening, morning. What is it? I don't know. I flew in from somewhere, but anyway, my name's Alex Boye. I'm not in the press or anything. I'm your entertainment for tomorrow at the hotel. But I really wanted to come, and because sometimes you don't know until you know. You have no idea. There's many people. Let me give you an example. I do a lot of charity work, particularly with just different charities in general, one suicide prevention. But a few years ago, I did one where I was invited to perform, and it was for polio, and it was for the total eradication of polio. And they were saying that, there was this big celebration. It was through the vaccines, and it was, and I remember just thinking, wow, it's pretty awesome. I want to be a part of that and learn there. So my question to you is, when we're thinking of the vaccines, especially with COVID, what is your what do you need to do to get this message out to people who sit on the fence, who say, no, I don't want to do this. As I said, I'm standing here and I'm listening to your words a lot that I don't understand. You know what I mean? But there is a lot. The messages that you're bringing out, I'm seeing the passion. I'm seeing your dedication, and I'm seeing your absolute desire to help. And you're literally, if we're in Superman terms, you're saving the world. You probably won't want to, you don't want to be seen as those people, but that's what you're doing. And so, what is it that? How do you get that message out to people who are sitting on the fence and saying, "Oh no, this is, I'll just wait it out," or whatever other answer that they feel that they need to produce. So,
Jason?
Yeah, in part by trying to do events like this. We've done a lot of Q&As, town halls, trying to address different populations that may have some hesitancies. I've had high school teachers in Michigan email me to Zoom in to their class, and I've tried to do that. Time is obviously limited. Kismekia has done a lot to address different populations and reduce hesitancy. So it has seemed that a group of vaccine hesitant people just need more information. And when they find out the facts that we've known about coronaviruses since 1931, mRNA was discovered in 1961, the lipid nanoparticles date back decades, it suddenly becomes this overnight vaccine that's been decades in the making. And it feels reassuring that it wasn't rushed. And mRNA has been in humans since the mid-2000s. It's been tested in clinical trials. So it's maybe the first time the public is hearing about coronaviruses, mRNA, but scientists haven't. And I think hopefully we've seen a little bit more of why we need perhaps a little bit more of scientists communicating with the public. So that way they hear about these advances earlier than at the last second.
thank you.
It's a little bit,
Drew?
it's even more complicated because there's a couple of issues. One of them is equity, so availability of the vaccine. And this is something I've spent a lot of time on. I've been working in Thailand since the spring of 2020, where we made our own Thai vaccine and we made our own production plant. And that was because their fear was they wouldn't get vaccine for years if it was made in the West. We've done the same in South Africa, where we built a production site and we're helping research there make their own vaccines. So getting vaccine equity across the world is important. Now, the problem with hesitant people is there are different kinds of hesitant people. There are people who are dead set anti-science. They don't care what we say to them because they think we're criminals and we should all be locked up. there are hesitant people that I talk to on nights and weekends to try and get them to understand what the vaccine is, what it does, and why it's so much better than getting the disease. So a lot of it is, it's not enough to just go to the scientists and say you need to convince the world. You need to figure out who these hesitant people listen to, and those are the people. Right-wing politicians need to say a lot more that the vaccine will save your life. All of them have received the vaccine, but on Fox News, that they all say don't take it. So it's a much bigger problem than simply hesitant people who are afraid of the vaccine.
I would add one more dimension to that and echo what's been said, which is this is not just hesitancy about vaccine. The United States, preventive measures, screening for certain diseases and testing is not uniform. It's not required, but we have many people who don't screen for high blood pressure or get their PSA checked or have breast mammography. So echoing what Drew said, diversity, equity, and inclusion are key parts of that have to be melded with education. And again, as well said, it has to come from people who are in those communities, who reflect those communities, and who can speak to why these are important and to counteract misconceptions or lack of education about why these things are necessary, not just for vaccines, but for general medical care.
One of the other learnings from the COVID experience is that social media can be a very powerful tool. In this case, unfortunately, it was for mis and disinformation, which had a negative impact on our ability to get vaccines. And then going forward, going to need to better understand the dynamics of that tool for positive purposes.
So now I would like to invite Douglas because as you, oh, sorry, excuse me. Sorry, absolutely sorry.
As scientists, you have been.
Who wants to start? Take this one.
And kind of bad for my liver. When we saw that the efficacy rate was 95%, I mean, I don't think anybody around this panel would say they couldn't believe that, So I had at least a couple of scotches at that point. And, and there have been various celebrations subsequently, but from a personal point of view, it's really quite remarkable. I mean, you go through your life doing what you do, and then you play a role in something as major as this. It's absolutely mind-boggling, humbling, that's for sure.
Phil, you may add something because I remember I called you in, I think it was January or February, just after the vaccine have been approved, and you were, even if you have been involved in the early development, you were very cautious still at that moment that it could be so successful, and
the results were so spectacular they were hard to believe, so this happens a lot in our laboratory. You get a spectacular result in the lab, and you're skeptical of it initially. We had that happen. The vaccine came out on December 16th, and we got 6,000 doses the next day at the hospital. And 6,000 people were vaccinated with that vaccine. Only about 10% of the people in the healthcare workers were positive for COVID and had an immune response against the COVID virus. But 1 month after they got their vaccine, 78% of them were protected with an immune response. That was unbelievable. But we got the next time point 1 month later, and it went up to 93%, and a month after that it went up to 99%. By then we were believers. was true, and wanted to let everybody
Is there any other question?
And just to follow up, what is the biggest thing we're going to see in the next few years out of all this?
In our emphasis, we have a vaccine R&D center, and so that's where we can put our resources on vaccines for infectious diseases. Where there's a lot of effort in viral vaccines now. We hear a little bit less emphasis on bacterial vaccines. But, bacterial infections, especially in the aging population, it's bacterial pneumonia is a primary cause of mortality in the older population. So I think there's a gap in our availability of bacterial vaccines. So have an impact in that area. We've only had, mainly we have 12 vaccines that all the kids get. And with the exception of one or two of those, they were all made around 1960 by Maurice Hilleman. And it's the same vaccines that all the young kids get. So there hasn't really been very much vaccine development since 1960 and up until now. And so suddenly, we have this brand new tool, and I think it's and we've never had a clinical trial like the one we've just had to show everybody that it's safe and effective.
To precise, I asked the numbers the other day. It's 2.5 billion doses of mRNA vaccine, not all vaccine, mRNA vaccine now, and there's about 1 billion people that have been boosted. So we have kind of, an understanding of what's going on. It's an enormous clinical trial.
Yeah, all those vaccines that were made back in the 60s, eventually the population got comfortable with it. We don't hear uproar about vaccination to the same extent that we hear about it with the coronavirus RNA vaccines right now. All pretty comfortable with getting their kids vaccinated with those early vaccines that were made. The flu vaccine takes for every dose, it takes one egg for one dose of a flu vaccine. When you make 200 million doses of vaccine, you need eggs. And, and then there's all kinds of problems. We've talked about manufacturing problems with that, and purity and egg allergies and other problems that come with it. So that's going to be history. Looking forward to an amazing.
Yes, please.
Thank you. Can you talk a bit about the challenge of a vaccine with lasting immunity, sort of beyond what we're seeing in the current shots? Can you talk a bit about some of the unanswered questions or obstacles that still remain in sort of solving that particular problem?
I'm not sure.
Could you
Which problem
restate
did
the
you
question? Could you restate? I didn't hear the first part.
Oh, sure. So looking at vaccines, in the future of is there a possibility to be able to administer a shot that can protect a patient for up to a year, or perhaps even longer? If you can explain what it is about solving that particular problem that still remains a challenge from a scientific perspective.
Okay, Jeffrey.
One approach to providing more durability is to have broader immunity against other proteins of the virus. Right now, almost all the vaccines are focusing on the spike protein for logical reasons. It's a target for neutralizing antibodies and has worked very well. But looking forward, if we want to have a vaccine that is not only more durable, but potentially preemptive, meaning protecting against strains of virus that might crop up in a year from now, we might want to target some other proteins of the virus that are more conserved across these different virus strains and elicit T-cell-mediated immunity as well, which engender T-cell memory responses, which can then be recalled when you're exposed to the virus at some point in the future. There's actually a growing body of evidence now in the scientific literature that T-cell-mediated immunity against prior exposure to some other coronaviruses is affording people protection. If we broaden the vaccines to target other antigens in particular, cell responses, I think we have a better chance of avoiding.
Yes, with that, I think we were just mentioning, Phil was just mentioning the vaccination of the frontline worker here in Irvine. And we have to remember that at the beginning of the pandemic, I remember the government gathered they were on Zoom, and they have this WHO massive operation where they were all saying that we are going to collaborate and we are going to vaccinate the population this way. We will start it with the aged population that is the most at risk and the frontline worker because they are the most at risk. And then after we change our mind and we say, okay, no, we are going to vaccine our population entirely, and we will see for the other country maybe later, which drive to the situation. But there was a big logic to protect the frontline worker because, the world would have stopped completely without all these people, be it at the hospital, but also be it, at your grocery store, people that were doing the delivery and etc. So this celebration is also about them, and maybe Douglas can explain why it is so important to have them included with this prestigious scientists. It's a collaboration of the two that make us out of the woods, sort of.
Well, first I want to thank Fabrice. You understand why I was looking for someone who can tie all these loose ends together and tell the story that are manifested by these men here and their colleagues. And Fabrice, you did a great job. My French is getting much better listening to you, so thank you so much. I also want to thank my panel here for coming. They are all extraordinarily busy individuals, and just two of them, Dr. Weissman and I think who else is leaving? Dr. Graham are going to be here tomorrow night, and then they take the red eye to Washington to a National Academy meeting there, where our Dr. Graham and perhaps Dr. Weissman are going to be honored by their National Academy peers. So it's amazing they flew out here for this event to be here with us, and that's extraordinary for me. I had mentioned earlier that this is a celebration of gratitude and certainly to these men and their colleagues, but also to the local community. If you have an opportunity to join us both for the panel this afternoon and for the dinner tomorrow, you will meet these men and women and young adults that have made our life much more comfortable, brought us back from, in some cases, we were terminal, and yet we lived because of the care that was given to us. And you'll have a chance to meet these folks. I've been asked many times, and I want to raise this question as a closing question. The question I was asked was, okay, Doug, you brought these scientists and these heroes together for this celebration. What happens when you're done? What happens if you have any money left over? What happens to the message that you've been creating here in Southern California? And that was an easy answer for us, because right from the very beginning, one of the things we wanted to do was to highlight both the science and the scientists and the courage and tenacity of our local community, but we wanted to have something that was going to live on after our celebration was over. And that goal was to help our healthcare community heal. Because of all the demographics that we see, two really stand out as the ones who have been suffering the most as a result of the pandemic: our children and our healthcare community. And what we decided to do was to focus on the healthcare community. How do we help those who helped us the most at the greatest personal sacrifice, not only to their own lives, to the lives of their own family, to their colleagues? I've had dozens of conversations with healthcare workers who slept in their car or on a mat in their office, never went home, or if they went home, they were not allowed to get anywhere near their family members. Some of them were terribly sick, and two that I know of died in the course of their duties. How do we help? And we turned to the mental health issue because the mental health issue affected even a broader group of people. Not every healthcare worker was physically sick, but the emotional trauma and the mental stress. I spent 10 years in the Air Force. I was during the Vietnam era and beyond. I have veterans in my company, and they know what PTSD is. And we know that our healthcare community is suffering. So we turn to an organization that will allow us to live on and help our community. And I want to introduce to you the person who's going to help us accomplish that. And that is Dr. Jonathan Goldfinger from the D.D. Hirsch Mental Health Services. Jonathan, just come up, give us a minute of what you're about to do. And so I want to pass the torch from the gratitude we are expressing to the resources that we hope will be amplified by what we're doing today to make a difference in our communities in the future. So, Dr. Gold. Thank you.
Thank you so much. Thank you for having me. I know I'm standing between all of you and a meal, so I'll be brief. And to all of you up here, it's an incredible honor to listen to your story and your journey. My journey as the CEO of D.D. Hirsch, an 80-year-old leader in whole person mental health and crisis care, was slightly different. I spent the last 2 years preparing for the after effects of the pandemic, both from the viral illness, where sadly some of the preliminary data does show it contributes to mental illness that we're seeing in the population, but all the traumas that Doug noted for young people and health workers especially. And so D.D. Hirsch is based out of Los Angeles and Orange County. We provide 24/7 crisis hotlines. Glad to see you here, Alex, one of our great ambassadors that we've been proud to work with. And so with the funds from these incredible events and really honoring all of you, thank you for being here and making this possible. Doug and Charlie, they've asked that D.D. Hirsch utilize the funds to provide newly developed, not as fast or as much NIH funding, I'll say, but newly developed support groups and suicide specialized therapy, individual and family therapy for health professionals, our frontline health workers. So we're going to be able to save their lives and give them a future for what they gave to all of us, thanks to your research and their heroism on the front line. So I'm honored to be here. I'm honored for our organization to do this for the health workers of Orange County with Doug's support and with all of you, and grateful just to have this opportunity. Thank you very much.
Well, thank you all. Thank you for all our prestigious scientists to answer your question and explain their journey. Thank you for attending. And with that, I think everybody wants to go to lunch. So, bon appétit.
And there, but before they had the step, it was,
That's right. This is yours. Thank you.
I did it again without falling.
Is that so?